Hey there, ladies and gentlemen, boys and girls. Today we're going to talk about the last type of ways molecules can move across that phospholipid membrane or a phospholipid membrane, taking advantage of that membrane's natural flexibility and ability to self-heal itself. And for those who were able to do the bubble lab, remember about how when you put things through it, you poked your hand throughout, it was still able to kind of close that gap before popping that bubble. And so we're going to take advantage of that today, and this is what we call bulk transport. This is what I like to think of the Costco method of doing this across. And by bulk, I mean large amounts, all right, or extremely large molecules. That's why I like to call it the Costco method, because you buy Costco, or if you've ever been to Costco or Sam's Club, you buy everything in very, very large amounts or very large objects to do this. And to do this requires ATP. So it is related to active transport in the fact that it requires an outside energy source, extra energy to do this, although we are not necessarily going from high or low to high concentration. So it's very related to it, or active transport in the fact that it both requires ATP. And so this is also going to take advantage of something called the vesicle. If you remember, a vesicle is just a small package within the cell. All right, it's actually a very small membrane-bound, phospholipid bilayer membrane-bound package that you can move molecules in and out. And so we're going to show the two different ways, or the two different types of bulk transport. All right. First one we're going to talk about is called endocytosis. All right. Endocytosis. All right. And this is a type of bulk transport where things are going to be entering the cell using a vesicle. All right, vesicle is that little package, remember, membrane-bound package. So let's say here, I'm looking at this one here, that black line is my phospholipid bilayer. Just kind of zooming out a little bit here. Might be a little hard to see, but I have let's say I have these, let's say these are amino acids. I just ate some food, I digested them, and I need to bring those amino acids in to help produce more proteins. All right, amino acids being the building blocks. All right, but I don't want to do like one at a time. I want to get this huge shipment in all at once. All right, and so what we're going to do is we're going to take advantage of that cell membrane's fluid shape and be able to bend. All right, so I'm just going to draw kind of like a little timeline of what's going to happen here. All right, first thing that's going to happen is that cell membrane, as that material starts to kind of come in, all right, it's going to start to bow in a little bit. All right, allows that material to kind of come in there into that area, all right, and then it starts to kind of close in on itself. All right, it's starting to kind of pinch off, and now it's got some of that material. We said amino acids inside. Now we're starting to form a vesicle. It's starting to pinch in here. All right. It's going to pinch in, and then once it closes that gap, we got those amino acids in there, enclosed in that. Now what we have is our vesicle. All right, now we have our vesicle here. All right, that vesicle can go off into the cell where it needs to go. All right, this is what we call endocytosis, because it is entering the cell using a vesicle. That membrane kind of wraps around the materials. Once it pinches off, once it kind of closes that space there, then it can pinch off and go off to where it needs to go. And now there's actually two different types of endocytosis, just kind of depending on what you're bringing in. There's either what we call phagocytosis, is what we're doing with larger molecules. Also known as cell eating, because think it was like eating larger pieces of food. Unicellular organisms like amoeba, this is how they eat their food. Instead of it being amino acids, it's maybe a smaller unicellular organism. All right, and they basically just take them in, and then they bring in enzymes to digest that. All right, and sometimes they're for smaller molecules or liquid molecules, and that's called Pinocytosis. All right, Pino meaning small. That's for smaller or liquid materials that you're trying to bring in. All right. So that's our endocytosis. All right. So now let's say I'm trying to get rid of a lot of stuff. I'm trying to ship out a lot of stuff. And that is the opposite of endocytosis. That is exocytosis. All right. So endo, I always like to think of as entering, E-N. All right. This is exocytosis. All right, exocytosis, which is, I'll just keep out that word there, exiting the cell with the vesicle. Kind of just opposites of each other. All right, so it kind of works in the same way. Let's say I have a vesicle here. Let's say I just ate a bunch of food, and I just ate a bunch of food, and my cell produced a bunch of insulin. And I need to release that out into the bloodstream so that they can attach to the glucose and to the blood sugar. All right. So I have a lot of insulin in here, insulin proteins. All right, and I needed to get it out into the outside that cell. What it's going to do is it's going to come up to that cell and it's going to attach. All right, once it attaches, all right, it's going to start to kind of open up. All right, those membranes of the vesicle and the cell membrane are going to attach. Kind of opening up so that in this case, the insulin can be released. And then the cell membrane kind of absorbs that that vesicle. All right. Now that insulin is outside that cell. It's gone through exocytosis. It's some kind of a bulk thing. So kind of think of this is, bends in, step 1, kind of folds in on itself, step 2, the vesicle pinches off, step 3. In this case here, kind of the reverse. Step 1, the vesicle arrives. Step 2, it merges with that membrane, opens up, starts to open up until it blends back into that phospholipid bilayer. All right. So the things you need to know: differences between endocytosis and exocytosis, and a little bit about how it looks when it gets done. All right. Otherwise, that's going to be it. This is actually hopefully a shorter one today. Thank you for watching. Good luck, and may the science be with you.